Okay, so I'm going to switch on to the genetics handout now. And does everybody have one? Okay. We'll be going on to flu after the break. So. Okay, so viral genetics. These are nucleic acid. These viruses contain nucleic acid. So the genetics is basically the same principles as all the other genetics, you know. Nucleic acids are nucleic acids. They're basically chemicals. The rules apply. But viruses have got some areas where I just want to focus on because they do things, they have some additional options, just like we saw with DNA synthesis. So the basic principles are the same. So a lot of this is going to be review, and I'm just going to go through it quickly. But I just want to stress a few areas where it's important to think about in relationship to viruses. And the genetics of viruses have been really important in enabling us to understand things like their pathogenesis and their life cycles. So it's been very important in developmental research areas. They're very important in understanding problems and with vaccine development. And also, of course, things like drug resistance are very important. And the more we understand about how these viruses behave, the more we understand about how treatments should be given, just as you've seen with antibiotic resistance and things in bacteria. Once you understand the basis of some of this, you can try and minimize some of the impact. Not terribly. Okay, so let's start with one thing which this is a quote, that here it says, the DNA chromosomes of eukaryotic host organisms, i.e. you, generally require geological time spans to evolve to the degree that their RNA viruses can achieve in a single human generation. So what they're saying is viruses change much more rapidly than do their hosts. And why is that? Well, one thing is to think about. Viruses grow very rapidly. So when a cell is infected, the first new virus particle may be out within an hour or less. And many of these viruses are lysing their cells by about 5 hours. You've got a huge amount of virus being produced, like 5,000 particles, 10,000 particles. So a single particle produces a lot of progeny. Now, if you think about it, by the time you've got to the end of my lecture this morning, if you were a virus, you could have produced 5,000 virus particles by the end of this lecture. And if all of those and you were producing another 5,000 virus particles by the end of the halfway through the next lecture, and if they didn't die of terminal boredom, they could do another round. But you'd have a huge number of great-grandchildren by 12 o'clock. So this is the big problem. Their generation time is incredibly short, and they produce an incredibly large number of offspring. So what you will find of these huge numbers that are produced, there are going to be lots of mutations, but they're just produced so quickly and in such huge amounts that this virus pool is going to be enormous. So that's one of the reasons why you get these really rapid changes. Now, you see this with the DNA viruses, huge number of potential mutants in these populations, but they have access to proofreading, so they're rather more accurate than the RNA viruses, as I pointed out yesterday. So the RNA viruses don't seem to have access to proofreading. We generally do not proofread our RNA, and RNA viruses never seem to have found a good method to proofread their RNA. The DNA viruses can either use our things, or they seem to snaffle our genes, probably, and then adapt them to their own purposes. So, in view of that, what you might expect is you'll see a lot of mutations can arise very rapidly in the DNA virus world, but they arise even more rapidly in the RNA virus world. So, therefore, when you come to look at the genetics, it's very important whether these viruses are RNA or DNA, because, as I say, DNA viruses tend to be somewhat more accurately copied and be somewhat more stable, but the RNA viruses can be very variable. And also, it matters whether they're non-segmented or segmented, and we'll go into why that is a little bit later on. So how do viruses change? How do they mutate? Same ways as we do: mutation, recombination, insertions, deletions. But I just want to quickly review mutation for a moment. So how do you get mutations? Same way as we do. You can get polymerase errors. These are often due, as I said yesterday, to the tautomeric form of the bases. And I've got a picture here. if you look at these are the standard form of the bases that you see in the Watson-Crick helix, but you can get a redistribution of electrons around the ring. And so what you have is the thymine, instead of having a double bond O here, has OH. And therefore, this H can participate in a hydrogen bond reaction by donating to something like a nitrogen or an oxygen. And it behaves like cytosine, which has also got an H in this position. And you have the same thing with all the other bases. Each purine behaves as if it was the other purine for a tiny fraction of time. Each pyrimidine behaves as if it was the other pyrimidine for a tiny fraction of time. If the polymerase incorporates them in this form, it will incorporate. If thymine arrives at a site where the polymerase is about to add a C, and it's in this form, it will be added. We have proofreading enzymes that would repair that because by the time it gets to proofreading thing, it's already flipped back into the true form, the normal form, the Watson-Crick form, and it will be seen as a mismatch and repaired. The RNA viruses don't have access to that. So you get the tautomeric form of basis. There's no way around it except proofreading because this is just a chemical equilibrium. There's nothing too much you can do about it. So however good your polymerase is, it's never going to be able to deal with that one. And as I said, we said the mutation rates are usually higher in RNA viruses because of their lack of proofreading. So one question here is, why, if you've got all this high mutation rate, why is it that if you vaccinate me against mumps or measles or rubella or rabies, I can travel the world and I'm still protected against those? In other words, these have got the same serotype wherever you go in the world. Why is it that they don't change? And it seems that what has happened is that every time there's a mutation, if it enables the virus to grow more rapidly and spread more rapidly, then that mutation will be favored in the environment because you're producing so many that anything that's better than the others will have a big advantage. So what's happened over the eons, since they've got such a short life cycle, you get these changes can be fixed very rapidly. So what happens over the eons is apparently these viruses are so adapted to their particular life cycle that if you make a change, as a major change in any of the proteins, or even a single amino acid change apparently in any of the proteins, they just are not as fit to compete with all the other viruses that are there in this pool. And so they don't produce as many offspring and they soon get outcompeted by the wild-type virus. So the circulating virus is called the so-called wild-type virus. Now, actually, if you sequence these viruses at the nucleic acid level, you do find there's some variation, but it doesn't lead to major variation in the protein. So you can find these, because of the degenerate code, you can find these changes. So in fact, the labs around the world who are trying to track, if somebody goes down with measles, where did this measles come from? It's known that the measles circulating in various parts of the world has got a different nucleotide here or there, and so you can look at the person with measles who's come into this country and see if they got it from somewhere in this country or whether their measles is consistent with them having got it wherever they've been traveling outside. So you can use nucleic acid variation to look at it, but at the protein level, these don't change their serology on the outside and they seem to be extremely conserved. And we think that's just because they have been so adapted to their host that any mutation is deleterious. So you can kind of think of this as a so-called fitness peak. They're kind of sitting on top of the mountain, and anything that changes that properties of that virus means that they sort of slip down the mountain and are less fit than the other ones. And so it's very difficult for them to change because, as I say, everything is less fit. So we think that's probably what's happening. Okay. So the origin of the mutations, they can be spontaneous due to things like tautomers. They can be physically induced by such things such as UV light, which is especially a problem if you don't have anything to repair. In DNA, it's the thymidine dimers. In RNA, you get uridine dimers. So when they're exposed to UV light, you get those dimers. And if you remember, in eukaryotic cells and prokaryotic cells, there are mechanisms to repair that kind of damage because it's very easily induced by UV light, and there's a mechanism to repair it. But again, the DNA viruses may have access to that kind of repair mechanism. Our cells don't provide anything to repair uridine dimers in RNA. If we get uridine dimers in RNA, it's just junk and it doesn't really matter. We regard RNA as very much a disposable item. So the RNA viruses are particularly susceptible to UV light, and they don't seem to have access to repair for that. So if you get a uridine dimer, the polymerase can't get past it when it's copying it, and it's the end of the story for that particular genome. X-rays, all the other things that would damage regular nucleic acid, damage viral nucleic acid. It is regular nucleic acid. And you can have chemically induced changes just like you can for our things. So things that modify the bases such that they behave inappropriately will also cause mutations. And again, you can have the standard types of mutation, point mutation, one nucleotide replaced by another, insertion where you actually insert a chunk of nucleotides, or deletion. So these mutations lead to changes in the properties of the organism. In the case of viruses, obviously changes in the properties of the virus, and changes in the properties are called phenotypes. And so what kind of phenotypes do you look for in viruses, and how can these be useful? So when you come to look at these phenotypic changes, which in this case is a consequence of the underlying genome change, you can see so-called conditional lethal mutants. And these are mutations which are lethal to the virus in some circumstances, but not in other circumstances. And a standard one of these is temperature-sensitive mutants. So, for example, a temperature-sensitive mutant might be able to grow at 31 degrees, but not at 37 degrees, whereas the wild type could grow at both temperatures. So what that would mean is that if you were to think of this in terms of the human, what it might mean is that a temperature-sensitive virus might be able to replicate in your respiratory tract, because that's a lower temperature than 37 degrees, but it won't be able to replicate inside, because that's 37 degrees, and that would not be compatible. Usually that seems to be because the protein that has this mutation in can't maintain its proper conformation at the higher temperature. So temperature sensitives are one example of these conditional lethal mutants, and these have been very useful in the development of vaccine. And host range mutants are also a conditional lethal mutant, but in this case they can't grow in all the same cell types that the wild type does. So you can grow these mutants under various conditions. You can grow temperature-sensitive mutants at the lower temperature, and you can grow host-range mutants in the right cell types, but they have a much more limited one. So one thing is, by looking at what mutation has occurred to stop them replicating in some cell types rather than others, tells us what they need from those cells that they're missing. So we start to understand how they interact with the cells. They're also useful in vaccines. So, for example, if you take poliovirus, it normally replicates in your gut, and it's not a big deal. But if it gets into the dorsal root of the spinal cord, it can be a big deal, and it can lead to paralysis. So if you had a host-range mutant of polio that could grow in your gut, but not in your spinal cord, it wouldn't be a problem. So these host-range mutants can tell us a lot about why these viruses are pathogenic, how they're pathogenic, and they can also help in things like vaccine development. I showed you in one of the earlier lectures that you can assay viruses because an infectious virus particle will kill a cell in a single layer of cells, and then its progeny will kill the cells around it, and you gradually get an area of dead cells, and these are called plaques. You can look at viruses and pick up ones which have got changes in them, because you can just screen a plate and say, you know, that's a really big plaque and this is a really small one, and then you can take the virus from those plaques and see if they breed true, and if they do, you've probably got a genotypic change which is affecting plaque size. That may seem very trivial, and it is in many ways, but what you sometimes find is the viruses that give smaller or larger plaques, it's unpredictable, have got changes which also mean that these changes, besides affecting plaque size, may affect pathogenicity. So it's a way to select a different virus, and then you screen them and see if it has any effect on pathogenicity. So they have been used. Drug resistance is something we're constantly worried about. and you'll hear stories of drug resistance already. We haven't been using antivirals for very long, but drug resistance occurs very rapidly, unfortunately. And you can get enzyme-deficient mutants, and we're coming across those again. Some genes in some of these virus particles are optional. The virus only needs them when it's in certain cells. One example of that is the herpes virus thymidine kinase. Remember, in the herpes virus lectures, I said that herpes virus provides its own enzymes for making deoxynucleoside triphosphates for making its DNA. And one of those enzymes is thymidine kinase, which enables it to rescue nucleosides and convert them to nucleotides and then use them. So it pulls those into the cell and increases the nucleotide pool. That is optional for herpes because if it's growing in a rapidly dividing cell, when our cells go into S phase, they make those deoxynucleotides. So it doesn't need those enzymes for deoxynucleotides in rapidly growing cells in tissue culture in the lab. They're there. But it does need them if it's going to grow in your neural cells because they aren't in S phase, and so it's going to need them there. So the herpes virus, its thymidine kinase is optional. If it's in cells that are cycling, it can use the sources from the cell. So you can delete the thymidine kinase and grow it in tissue culture, but it won't necessarily be able to grow in all cells. There are also hot mutants, so-called hot. They're reverse of temperature-sensitive mutants. They grow better at higher temperatures. And these can often be more pathogenic because our fever response is something that helps us to, very frequently, these viruses under fever conditions seem to grow less well. They don't like temperatures much higher than 37. So these hot mutants, if they grow better at an elevated temperature, may be less susceptible to the fever response. And there are attenuated mutants, and they may be attenuated for all sorts of reasons, because they're temperature sensitive, but attenuated mutants refer to their pathogenic properties, and they tend to cause either milder symptoms or no symptoms. These are obviously important in vaccine development. If you can get a mutant that causes no symptoms but has got the same serology, it protects you. And they also enable us to understand pathogenesis, because why don't they cause symptoms? We can look at the changes and see what it is that's important for pathogenesis for these viruses. Okay, any questions there? So, mutation is one way that you get genetic change, and another way that you get genetic change is recombination. And recombination is a simple description of it is it's the exchange of information between two genomes. And we obviously have DNA recombination. The viruses can use some other methods that we don't regularly use, so I'm just going to go quickly over those. So classical recombination of the sort that we use in E. coli, etc., is that there's a crossover between one genome and the other. This occurs at areas where these two genomes have got similar sequences, so this is homologous recombination because the sequence here is the same as the sequence here. Sequence here and here are the same, sequence here and here are the same. So you get homologous recombination, and the result of that is that this region gets inserted into the pink bit gets inserted into the blue and vice versa. And that's how we do it. And in fact, many of the DNA viruses have similar recombination type mechanisms, and they may use our recombination mechanism or they may use their own. But when you come to RNA viruses, we don't have a recombination method for RNA, and the RNA viruses, again, don't seem to have developed their own. There doesn't seem to have been even any starting material because, as I say, other organisms regard RNA as being a disposable item. So for the RNA viruses, you're going to see something different, but the end result is going to be that the pink can give something to the blue or vice versa. So this is common in DNA viruses, but as I say, you don't see it in RNA. So how can RNA viruses get around this problem if they are going to do recombination? Well, one thing that you see in quite a few of the positive strand viruses, and the picornavirus includes polio group is an example of this, is something called copy choice recombination. So if we take two strands of say a picornavirus here, this is one of them, this is another, and we're just imagining that this has got a mutation here, which is lethal to the virus, so it can't grow very well. And this strand 2 has got a mutation here. So if you've got these two viruses, how can you get a recombination that would give you a thoroughly viable virus? And one thing that seems to occur is during RNA synthesis, and I'm not showing the VPG here at the end or anything, but during RNA synthesis, the RNA will be copied in this direction, and you'll have a growing strand of RNA, and it will actually be predominantly single-stranded. It's not made as this double-strand intermediate, but as it went past the green, it will copy the green gene into the good form, and then it will carry on. But what seems to be able to happen in these viruses, I didn't tell you, but this replication in poliovirus all occurs in a highly organized fashion on membranes in the host cell. So they're held together very closely. So each replicating molecule is very close to the next replicating molecule. And it seems that what happens is that this growing molecule here can flip across, which causes this negative strand, can flip across to this positive strand and bind to this just as well. It's only probably got one base pair difference, and that doesn't matter. So it leaves this strand and joins this strand. And now it matches perfectly along here. It will have Watson-Crick base pairing here, and it will just extend here and copy this one. So, of course, when it gets to this gene, it will copy the good form of this gene. So what you end up with is a negative strand which has got the good version of both genes, and when that's replicated, it will be replicated into a positive strand, which has got the good form of both genes. So now basically what you've got is recombination, that this positive strand here is given the right-hand good version of the gene, and this strand here is given the left-hand good version of the gene, and you've got a recombinant. So this method is called copy choice, because first of all the polymerase chose to copy strand 1, and then it went over and copied strand 2. Any problems with that? It's pretty simple. It's just you have to think about it a bit. Okay. So we see that in quite a few of the negative strand viruses, of the positive strand viruses. What about the negative strand viruses? Do we see it there? And the answer is we don't see it in the negative strand viruses. When I went through the negative strand virus life cycle, I said they remained as nucleocapsids. And in fact, it seems that most of the cytoplasmic RNA viruses, and the nuclear ones, remain as nucleocapsids while they're being copied, because their polymerase can work with it, and RNA is pretty fragile, and it's a good way to protect the RNA when it's in the cell. So they work with a nucleocapsid. So when they're copying the nucleocapsid, if this RNA is dissociated from this one, when it hops across to this one, the chances of it being able to hybridize and act as a good primer for making a continuous negative strand are very remote, because all the bases are hidden underneath the nucleocapsid protein, so it can't get at it. So you don't see this kind of recombination in the negative strand viruses, and we think that's because, as I say, they tend to be in nucleocapsid form, and it's just difficult for this RNA to find the right region to prime off on this strand. So it just floats off. If it comes off, it just floats away. Okay, no problem? Okay. So there are other methods of recombination, because as you doubtless realize by now, these RNA viruses, there's probably quite a few simple solutions to these problems, particularly if you have got so many RNAs in the cell that if something goes wrong, it doesn't really matter. So they've got lots of possible solutions, and a lot of them take advantage of these RNA viruses, as we've seen, have their own quirks during replication about what they can do, and so these other types of recombination take advantage of those. But they basically many of them work on the same idea as copy choice, but they may have some different details, just because, as I say, each of these seems to take advantage of the quirks. So some of these RNA viruses actually have very high recombination rates. Many of the coronaviruses do, and it's a kind of variation on copy choice, but it's very efficient because it takes advantage of a particular quirk in their life cycle, and that includes the SARS virus. So that's always one concern about these viruses. That was this virus that appeared a few years ago and raised a lot of concerns, and fortunately has gone we have seen very few cases since. But a virus that can recombine very rapidly is potentially going to mutate, and it's concerning about antiviral things. So how do we use recombination? One thing is we can map genes on these viruses, because the closer two genes are, the less likely you are to get recombination between them. So it's just the same way as you would use recombination mapping in regular genetics. That was very widely used until relatively few years ago. But one of the things is, with the advances in this Human Genome Project and the sequencing developments that went along with the Human Genome Project, sequencing tends now to be so much easier than mapping that most viruses these days are sequenced rather than bothering to map genes by a rather long and tedious method. So, for example, when SARS was first discovered, I think it was less than a month before we had the sequence for it. I mean, it's very impressive how you can take an unknown virus and within a month you know what proteins it can code for and have a huge amount of information. So that's sort of one byproduct of the Human Genome Project, that the amount of sequencing and the ease of it has really improved rapidly. But it used to be mapped by recombination frequency. It used to be mapped by marker rescue, and I'm going to go into marker rescue because it's basically a principle that is used. So let me just explain how you used to do marker rescue. If you wanted to know where the thymidine kinase gene was on HSV, as an example. So here you've got a mutant gene. This particular HSV makes no thymidine kinase. No problem, growing tissue culture perfectly well. So if you were to take a wild-type HSV, herpes simplex virus, and fractionate its genome into chunks with, say, a restriction endonuclease, you could isolate each chunk, and then your question is, which chunk corresponds with the thymidine kinase? And what you can do is you can take this chunk, and you know where it maps on the genome, but you're asking, would it be this chunk that would be thymidine kinase, this chunk, etc.? So you take each chunk individually, and if it's the right one, which is the only one I'm showing, you'll get homologous recombination. And then what you will find is that the wild-type thymidine kinase will become inserted into the mutant virus. And now this will be positive for the herpes virus thymidine kinase, and you know that that therefore coded for it. That's called marker rescue. We use the same principle to insert all sorts of genes into viruses, and that's used in things like gene therapy, vaccines. So this same idea of replacing this by something else. Let me just show you that. So here, instead of adding the thymidine kinase gene, the wild-type thymidine kinase gene, this is a construct that has been made in the lab. It's got the ends of thymidine kinase gene on either side, so you can get homologous recombination. But inside, it's got the rabies virus G protein. So when this recombines, you now get the rabies virus G protein inserted into the virus. And in this case, I've put vaccinia virus as being the DNA virus that's used rather than herpes. The reason I actually chose this is, theoretically, this could be used as a human vaccine, and it hasn't been so far. But it is used as an animal vaccine. So one thing is that rabies is carried heavily by raccoons. And one thing is that you can try and stop it in the raccoon population by putting out a bait that has got an animal pox virus with a rabies virus gene inserted into it. They eat this, they get a minor the virus grows. It doesn't cause any symptoms. So the virus grows, and it makes the herpes virus, the rabies virus G protein that's put in the host cell membrane, and so this little raccoon will develop antibodies to rabies. It's been widely used in Europe to try and stop the spread of rabies in foxes. It's used in this country to try and stop the spread of rabies in raccoons. So these kind of things have got uses, and these kind of approaches are also being used for gene therapy, and there are also interests in trying to develop certain vaccines that will have these kind of recombinant viruses. As I say, so far there are no human ones that are based on herpes or vaccinia. Okay, so you can use recombination for the DNA viruses. Something else, though, there's a final form of a sort of relative of recombination that I want to talk about, and that's reassortment. And this applies to viruses that have got multiple segments. And for us, the viruses that are important here are all RNA viruses. So I said influenza, rotaviruses have got the multiple segments. So their genome consists of more than one segment. So if you took a purple virus and a green virus, and these are basically the same virus, but this has maybe got a mutation in that makes it more or less pathogenic or whatever, something that you can detect. If you put these two into the cell, then this will make purple copies of its genome, and this will make copies of the green genome. But when you come to package them, as far as packaging goes, it will be random as to whether you get two purple, two green, or a mixture of purple and green. So these viruses are said to be reassortments because they got some of each of the input genomes. And this is really efficient because you can see, the cell is just full of copies of purple and green, and it's going to be just random as to which gets into each of these. So what you can make is reassortment virus particles. And so these are basically recombination. You've got some of the genetic properties of this are being exchanged with some of the genetic properties of this. So this is a form of recombination. It's very efficient, but it only occurs in segmented viruses. So you see this hugely in the true influenza viruses, but not in the paramyxoviruses because those got a non-segmented genome. Influenzas have a segmented genome. And it's being used in some new vaccines. There's a vaccine that's just been approved in this country for rotaviruses and a vaccine for influenza virus that was approved about 3 years ago now. And they're both based on these reassortments. So here's the one for influenza. It's called FluMist. It's, as I say, been on the market for 2 or 3 years now. It's based on an attenuated virus, which is shown here in grey. And this virus is cold adapted. In other words, it grows better at lower temperatures than higher temperatures. What that means is that it will grow better in your nose than it will in your lungs. So it's more likely to cause a mild upper respiratory tract infection than a serious lower respiratory tract infection. It's temperature sensitive, which also means that it's not going to grow very well in your lungs. So it's cold adapted, so it grows well up here, and it's temperature sensitive, so it grows even more poorly down here. Well, my lung doesn't go quite that low. And it's attenuated, even if it doesn't cause much in the way of any symptoms. And it's given as a live vaccine, which means you get mucosal immunity, you get IgA, which is a much better form of protection than IgG, which is what you get from the killed virus. And you deliver it by aerosol into the nose, so you get an intranasal delivery, replicates in the nose, you get a good IgA response, you also get an IgG response, and so you get protection against the viruses, and as I say, this was approved. So how does this work? This virus here has got the hemagglutinin and neuraminidase that this virus particularly has, and what happens is each flu season, what you do is you make a reassortment with the current flu virus, so that the reassortment has got the hemagglutinin and the neuraminidase, the two outer proteins, of whatever influenzas are circulating. At the moment, there are 3 strains that we make vaccines against. So this will have they'll make 3 different derivatives of this, reassortments, each having the hemagglutinin and neuraminidase of each of those 3 strains. And then what you have is this virus here has got the internal proteins of this one, and the pathogenesis is to a great degree controlled by those. But it's got the external proteins of whatever's circulating. So the antibodies that you raise for this will neutralize this year's influenza virus. So what you can do is you can readily adapt it to whatever strains are circulating. So a reassortment vaccines, 5 years ago I used to have to say this is a good idea. Now we have 2 of them on the market. Okay, so for the non-segmented negative strand viruses, there's no classical recombination. There's no copy choice. There's no reassortment because they got single non-segmented genomes. And so the non-segmented RNA viruses undergo the least amount of exchange of genetic material. So you have each virus according to the type of its genome, whether it's DNA or RNA, and the type of genome, whether it's segmented or non-segmented, and the type, whether it's positive or negative, those all seem to control its variability in the environment. So that's why I've been stressing the importance of some of these. Okay, that's a good place to take a break, so we can take a 10-minute break.